Hey grade 7s, it's Friday, as I like to say, Friday. What are you guys up to this weekend? Or is that too much of a lame joke? Sorry, I know what you're all up to this weekend, but find something special to do in the confines of your own home. Right, let's get started with today's lesson. Right, we are looking at separating mixtures, part 2. Yesterday we did very simple separation techniques, and today we are looking at distillation and chromatography. Good morning, I am Mrs. Hall. The learning objectives for today's lesson are as follows. Understand how mixtures can be separated, to identify scientific apparatus, distillation as a separation technique, to recognize the everyday uses of distillation, chromatography as a separation technique, as well as everyday uses of chromatography. I hope I inspire you today and encourage you to pay attention because you might just learn something new. Right, our first technique that we are looking at today is one called distillation, and here's a close-up of the new apparatus that we are going to look at in a lot more detail. This is called a Liebig condenser, and it is used in the process of distillation. Let's start with the definition of distillation so you get a clear understanding and where to from here as we build on those knowledge blocks. So the process of separating a solution by boiling it so that the solvent evaporates and then cooling the vaporized solvent so that it condenses. That is distillation. And for those of you who can't remember, condensation is changing from the gas or a gas back to a liquid. Distillation, very important, allows us to collect both the solvent and the solute at the end of the process. Let's take a look at the apparatus that you already know. You already know what a Bunsen burner is, what a thermometer is, and what a glass beaker is. By the end of today, you are going to know what a round bottom flask is, or a distillation flask. You are going to know what a Liebig condenser looks like, as well as a clamp and a clamp stand for those of you that haven't used those apparatus yet in your classrooms. Right, let's start with our actual apparatus. So over here we have our thermometer. I'm not going to explain the process to you yet, just take note of the apparatus we are using. So you have your thermometer, it goes in through the mouth of the flask with a cork in to prevent the water vapor from escaping. This is our distillation flask or our round bottomed flask. This is our clamp that holds on to the round bottom flask, obviously because it's got a round bottom, it can't stand on its own, it needs help from the clamp. The clamp is attached to what we call a retort stand, right? Then obviously these labels we will go through in more detail later on, that's part of the process. Here we have our Bunsen burner, which you know. Here we have our Liebig condenser, and I want to explain this in a little bit more detail. So we have an outflow pipe and an inflow pipe. So our inflow pipe is where the cool water comes in. So a rubber tube or little rubber hose is attached to the Liebig condenser and attached to a tap in the science lab, and the tap releases cold water to the outer tubes of the Liebig condenser. Where does that cold water go? It goes out through the outflow pipe over there in the basin in a science lab. So the whole point of the Liebig condenser is to cool down the inner tube of the Liebig condenser, and I'll explain that to you in more detail now. Okay, and here we have our receiving flask, or our flat bottom flask if you'd like to call it that, where we get all of our clean distilled pure water. Let's get started. So as you know, we set up our apparatus and I showed you how it was all attached and how it looked in the science lab. So let's start with our solution of salt and water in our distillation flask. This is a solution and we want to separate the salt from the water. And we're going to use a process called distillation. This is where we first use the process of evaporation followed by condensation. So we first of all light up the little Bunsen burner, apply the heat so that the solution starts to boil. Now the water will start boiling at 100 degrees Celsius because that is the boiling point of water, and when a liquid changes from a liquid into a gas, there you have your water vapor. Now naturally the water vapor, as the water is boiling, wants to escape upwards, but it can't because we've put the rubber stopper there. So the water vapor is now forced to go through this little glass tube here. We also have the thermometer here to make sure that we're at boiling point. So the water vapor can't escape out, goes through the little glass tube over here and into the inner tube of the Liebig condenser. So that is the water vapor traveling down the inner tube. The outer tube has the inflow pipe of the cooling water in, remember, attached to a little rubber tube or hose pipe in the lab, and the outflow pipe, that's where the cooling water goes out. So the water is constantly flowing around the outside of the inner tube of the condenser where we have the water vapor. Now what happens to water vapor when it's cooled? It condenses and changes from a gas back into a liquid. And that is exactly what happens here. Condensation takes place and the vapor comes out as pure distilled water. So we have separated the salt water solution by having the water vapor go through the process of condensation back into a liquid, but we have left the salt behind in the glass beaker over here. Right, so at the end of the experiment, you'll actually see little white crystals at the bottom of the distillation flask if you leave it going for long enough. And that is the process of distillation. Right, why do we have distillation grade 7? Why do we need to know about it? Why do we have to separate a solution like that? The distillation process is used to separate components based on their boiling points, and it's very important in everyday life, and you'll understand why now. Chemicals like common gas, diesel, and jet fuel achieve their boiling points at different temperatures. So, gas, petrol, jet fuel all boils at different temperatures and starts to evaporate. So what we do is we take crude oil, right? And we put it into our burner, and at different boiling temperatures, a specific type of product is condensed and becomes a new product. So through the process of evaporation, that water vapor is condensed and formed back into this kind of liquid over here. So for example, at 40 degrees, we get petrol for our cars. At 110 degrees, we get a special chemical. I don't know how to pronounce it, in Panthe. At 180 degrees, we get kerosene, which is used for jet fuel. 280 degrees, we get diesel oil. So through the process of distillation and just using crude oil and making it reach different boiling temperatures, we are able to separate so many different products that we use in our everyday lives. We also use it in desalination plants. So recently in Cape Town, with the huge water crisis and the water shortage, we had to set up a desalination plant and we had to change seawater into pure drinking water. Okay, a very expensive process, but can be done. So basically, salt water is turned into fresh water through distillation. We first take the solution of salt water, evaporate it, goes through the process of condensation, and we are left with pure water. The salt crystals are left behind. Alcoholic beverages are also made through distillation. So the alcohol is boiled off from the rest of the mixture and collected in a concentrated format. So you've all heard of refined gin and distilled gin. That's how it's made, through the process of distillation. We also use it in the medical field because distilled water is so pure, it is a common tool used in the medical field. Hospitals use distilled water to prepare food for their patients and sterilize materials. Surgeons use distilled water during medical procedures. So I hope that makes a lot more sense to you now, and you understand the importance of science and why learning it is so important. Let's take a look at chromatography. Now, chromatography is a method used to separate a mixture in which different parts of the mixture move at different speeds through a medium. Now, this is quite detailed, this explanation, so I've broken it down into little chunks for you, and then I will simplify it for you through my explanation with my diagrams. So let's first understand what it is. So, pigments are special substances that give color to living tissues. So certain materials like paint and ink, they are responsible for the red color of blood and also the color of dyes. Those are pigments. Right, let's go back. What are we trying to do? We are trying to separate a mixture in which different parts of the mixture move at different speeds through a medium. So we want blood to move at a different speed, or paint and ink to move at a different speed. But why, you may ask? We use chromatography to separate a mixture of different pigments. Some pigments dissolve more easily than others. We add a solvent, such as water or alcohol, to a mixture of pigments, and we let the solution travel through a medium, such as paper. The more soluble pigments travel faster and cover a longer distance. Okay, this is where the science is coming in. The less soluble pigments, in other words, the pigments that don't dissolve as easily, travel slower and only cover a short distance. In this way, we identify the different colors of the different pigments. So, what about everyday life? What if? What if someone crashed into your car and drove off, leaving scrapes and paint samples on your car? You'd want to find out who that person was, definitely. There are 3 potential suspects. Can you identify the car that crashed into yours from the paint sample and left on your car? Chromatography. It is a really useful method for separating mixtures for analysis, especially with forensic science. Different types include gas chromatography, paper chromatography, column and thin layer chromatography. But at our level, we only need to know about simple chromatography. But hopefully, as you get older and get to university, you're going to want to study all the other different methods of chromatography. So a mixture, such as ink, will separate in a solvent, such as water, into its components, producing as many different spots as components. If it is pure, it will produce just one spot. Let's take a look at our apparatus. So, if we were going to be doing this in class, we would have our 500 ml beaker over here. We would have our chromatography paper. I used to just use filter paper. We would have 4 different black pens because I like to let you see the differences in the different inks and it's fun. Okay, but you can do this with just one black pen. You don't need lots of different ones. Water, which is our solvent over here. Our splint or our pencil. This is to pin our chromatography paper onto the pencil or the splint so that it hangs into the beaker as shown in the diagram. We need a paper clip or press stick to attach the paper onto the pencil, and we need a pencil and a ruler, and I'll tell you why now. So let's look at our method. We're going to draw a pencil line across a piece of chromatography paper, approximately 2 centimeters from the bottom. We are going to draw a spot from each pen on the pencil line too, not too close together, or just draw one spot, that's fine. You're going to fold the top of the paper around a wooden splint or pencil and fasten it with a paper clip. Just press stick it if it's easier. You're going to make sure that the bottom of the paper is close to the bottom of the beaker, not touching though, and you'll see why later. You put enough water into the beaker so it touches the bottom of the paper, but it must not reach the pencil line. You then watch and wait. Right, so here's the start of our experiment, exactly like we went through our methodology and what we had to do. There's our solvent in our beaker, right? Our paper is not touching the bottom of the beaker. There's a 2 centimeter pencil line drawn where we put our ink spot, our beaker, our paper, and then obviously attached to our pencil or piece of wood at the top of our beaker. At the end, the water is going to be absorbed by the paper and it's going to move upwards. And as it moves upwards, it's going to separate the solute, the ink here. And the more soluble ink colors, remember, will travel faster up the paper, and the less soluble will not travel as fast, and they will stay behind over here. So I hope you're understanding that now. But why? Why is it important? You're going to see now. Look at our results. The pens will have different mixtures of colors to make the specific black color. And again, the yellow was the most soluble pigment. Okay, it moved the fastest. Did you understand what I just went through? I hope so. But let's go through a couple of generalized questions that you would be asked in this situation. What solvent did we use? Scientific terminology, solvent. We used water in this case, but you can also use something like alcohol. Why was the line drawn in pencil? Think about it. Okay, think we used black ink, right? We wanted the black ink to dissolve. Why was the line drawn in pencil? So that it didn't run into the pen dots. Okay, because then it would have its own pigments and its own ink that would run, and it would give you the wrong answers to your experiment, because you wouldn't be looking at the black ink, you would be looking at the line that was drawn across the paper. Why did the pens have different colors or different patterns? Exactly, but as I said, you didn't have to use all the different colors, just the one, but there are mixtures of different solutes. So your specific black dot would have its own mixture of specific solutes inside of it. Where were the solutes found? Okay, we're using the black ink. It was in the inks. Okay, in the different ink dots that you made. Why might a spot not move at all? Okay, why would some of the spots not have had any solute coming out of it? Okay, any movement. The ink may not dissolve in that solvent. Something to think about. Okay, not all inks are soluble. Why now do we have chromatography? Why do we have to learn chromatography? What is its use in everyday life? This is exciting, and this, grade 7s, is why I love science. Right, we use it for forensic testing. Chromatography is also used to help catch criminals. In line with programs like CSI, gas chromatography is used to analyze blood and cloth samples, helping to identify criminals and bring them to justice. It's clear to see that chromatography is an unsung hero when it comes to keeping you healthy and safe. So for those of you who want to be forensic scientists one day, you need to study science. You need to know what chromatography is. It is also used in drug testing, as chromatography can accurately identify substances within the bloodstream. It is widely used in sport to test athletes for doping or performance-enhancing drugs. Something to think about the next time you're watching your favorite sport. You've all heard of the term doping. It happens, okay? It's in the news often. How do you think some of these top athletes achieve these amazing results that no other human has ever achieved before? They're using performance-enhancing drugs. It's known as doping. It even happens in some of our high school sports. Some light humor. Okay, let's see if you get these cartoons. Remember the tortoise and the hare? Right, look at this one. The tortoise beats hare subject to performance-enhancing drug test results. How on earth did the tortoise manage to beat the hare? Okay, and then this one I love. Football players in the olden days, he's saying, man, do I miss the pre-game coin flip when they used to just flip a coin to see which side was going to start first. Now they have to go and urinate in a cup and get tested before their game begins. They don't just flip a coin and start. Okay. Right. Chromatography is also used for creating vaccinations, and that is so important right now. So here you can see this is the Ebola virus. Okay. But chromatography is useful in determining which antibodies fight various diseases and viruses. Scientists use chromatography in the fight against the Ebola virus, which was responsible for thousands of deaths, grade 7s, not so long ago. So they use chromatography to develop the experimental immunization, or the ZMapp. Process was used to find out which antibodies are the most effective at neutralizing this deadly virus. So interesting to think, how are we looking at the coronavirus right now? Are we using chromatography to find a vaccination? For those of you who don't know what the Ebola virus is, just a quick little infographic here. The source of the Ebola virus comes from infected bats, which are thought to transmit the disease to humans, or indirectly through other animals, which are then hunted for their meat. So something very similar to what happened with coronavirus, coming from animals and then transmitted to humans. Okay, they have a vaccination for this now. I didn't go into the signs and symptoms of Ebola. It's very graphic and quite horrific to look at, but very interesting for those of you that want to study medicine one day. Go and do some research. Right, grade 7s, I want you, if you want to, not compulsory though, if you have some time on your hands this weekend in the confines of your home, set up a little chromatography experiment. Take your black inks, make your little dots, follow the method I've shown you, and get some lovely little chromatography experimental papers hanging outside on your lawn so that you can see which of your inks had more solutes and which of the inks had solutes that were more soluble than others, the ones that traveled faster. Just something you can do at home. Very, very easy. You have all the apparatus you need. I want to end off with science. It's like magic, but real. Because hopefully, grade 7s, you've learned something new today and you've realized that science is actually interesting. And it's not that difficult. It's actually quite easy. It's something that can lead to so much more later on in life if you just apply yourself, stay focused, and do the extra research for things that interest you. And you will find out that you can go places with science. And who knows, you might just find the next vaccination for the next dreaded virus that hits us. Grade 7s, have an amazing weekend. Enjoy your weekend. Take a well-deserved break. You've had a busy week. You've learned a lot. I hope you found it interesting. I hope you've enjoyed it. I will see you on Monday morning.